Spinocerebellar Ataxia Type 3 is a polyglutamine disease, which are a group of genetic neurodegenerative disorders. They are caused due to expansion of the CAG repeats in diseased genes, wherein CAG is an amino acid code for glutamine or Q. This presence of an elongated glutamine tract in the coding regions of genes produces an abnormally long version of the enzyme and causes the enzyme to fold into the wrong three-dimensional shape. This leads to neurotoxic phenotypes such as loss of motor and cognitive function and neuronal degeneration. The gene for Spinocerebellar Ataxia Type 3, or SCA3, is called ATXN3, which codes for the Ataxin-3 protein. It is located on the long arm of chromosome 14 in humans. SCA3 is caused due to ACAG triplet repeat expansion of 55 to 84 repeats in exon 10 of the ATXN3 gene. This leads to the atrophy or shrinking of the cerebellum, leading to neurodegeneration. Ataxin-3 is involved in a mechanism called the ubiquitin proteasome system that destroys and disposes off damaged or excess proteins. A molecule called ubiquitin binds to these excess proteins, taking them to be degraded within cells. Ataxin-3 cleaves ubiquitin from these excess proteins just before they are degraded to use the ubiquitin again. Due to this cleavage of ubiquitin by Ataxin-3, it is known as a deubiquitinating enzyme. ATXN3 gene contains a N-terminal Josephine domain, two ubiquitin interaction motifs, UIMs, a third putative UIM in the C-terminal end, and a poly-Q stretch in SCA3 patients. The first two UIMs act as a polyubiquitin binding domain, allowing for a K48-linked polyubiquitin chain on Ataxin-3. The third UIM does not play a role in this. The Josephine domain is used to cleave the long polyubiquitin chains. However, in SCA3 patients, due to the increase of CAG repeats in ATXN3, a non-functional Ataxin-3 is obtained which cannot remove ubiquitin from excess or damaged proteins. Hence, these proteins, along with ubiquitin and Ataxin-3, form aggregates within the nucleus of cells. SCA3 is the most common autosomal dominant inherited ataxia in the world. However, at present there are no therapeutic solutions for this debilitating neurodegenerative disorder. One possible solution involves the use of antisense oligonucleotides, or AONs. AONs are short, single-stranded sequences of nucleic acids that can bind to a exon or intron splicing enhancer to mask the target exon, so that on translation the targeted exon is ignored. AONs can be used in SCA3 to target and skip exon 10 of ATXN3, which contains the expanded poly-Q repeats, while maintaining the reading frame. This redirection of the splicing of Ataxin-3 pre-mRNA allows the treatment of the cause of the disease. To test this novel approach, 5 splice-modulating AONs were designed. As shown by RT-PCR, 3 truncated Ataxin-3 proteins, that skipped exon 10, were produced by AON 10.1, AON 10.1-2 and AON 10.4. The wild-type Ataxin-3 pre-mRNA contains 361 amino acids. The antisense oligonucleotides allow skipping of exon 10 by binding to it. However, due to the presence of a stop codon at the start of exon 11, the Ataxin-3 pre-mRNA is truncated and lacks the polyglutamine repeat in UIM3. To test the feasibility of this approach in vivo, 4 antisense oligonucleotides were designed to target exon 10 of human ATXN3 gene with 84 CAG repeats. 500 micrograms of the 4 antisense oligonucleotides were injected into a mouse model as a single intracerebroventricular bolus, meaning a fast one-time injection. This was done by implantation of a cannula into the right lateral ventricle of the mouse. After 2 weeks, the efficacy of antisense oligonucleotides was assessed using Sanger sequencing. This demonstrated that as a result of exon 10 skipping, there was a stop codon, TGA, at the start of exon 11, resulting in a truncated ATXN3 RNA found in the cortex and cerebellum. To find out whether the AONs are effective for a long term in the mouse brain, 27 mice were cannulated and injected with 500 micrograms of AON 10.4, at 2.5 months of age. Additional 250 micrograms was given following that on week 2 and week 3 after surgery. resulting in a total of 1 mg antisense oligonucleotide per mouse model. The mice were then sacrificed 3.5 months after surgery. RT-PCR analysis demonstrated that following sacrifice of the mice, a 40% Ataxin-3 modification was seen in the brainstem and cortex, while a 20% Ataxin-3 modification was seen in the cerebellum. This indicates that the antisense oligonucleotides had distributed throughout the brain and are stable and effective for at least 2.5 months post-injection of 1 mg AON 10.4. It is also shown that the modifications of Ataxin-3 are dependent on the dose of AON supplied. For instance, for the cerebellum, with a 500 micrograms dose of AON supplied, there was a 14% protein modification, while with a 1 mg dose of AON, there was a 20% protein modification. Mutant Ataxin-3 containing the poly-Q stretch causes nuclear accumulation that leads to neurodegeneration. Prior to treatment, the mouse model showed a strong nuclear localization of Ataxin-3 in substantia nigra. However, following AON 10.4 treatment, the mice showed a decrease in the Ataxin-3 nuclear accumulation, which was possibly as a result of the decrease in the amount of mutant Ataxin-3 containing the poly-Q stretch present in cells. Thus, the injection of antisense oligonucleotides is able to reduce Ataxin-3 nuclear accumulation and insolubility. Overall, the removal of exon 10 in ATXN3, meaning the removal of poly-Q repeats, is likely to treat the cause of the disease and not merely the symptoms. Thus, it can restrict further neurodegeneration and thereby delay the development of SCA3. Clinical trials, conducted in 2016, involving injection of AONs, to treat spinal muscle atrophy in children have shown that AONs have a half-life of 4 to 6 months in the cerebrospinal fluid. Hence, extrapolating from this, a relatively infrequent dosing of AONs every 6 months in order to treat SCA3 could be a feasible treatment. Thus, the antisense oligonucleotide treatment serves as a therapeutic solution to Spinocerebellar Ataxia Type 3 and holds the potential to stop the progression of the disease completely. However, its efficacy is yet to be tested in humans.